Okay, so thank you. Hi, my name is Alicia Rodríguez, and I'm going to present a little work of my lab in the Astrobiology Center in Spain. And with your permission, with the experience yesterday, I'm going to start. Okay, so the work, I'm going to put the timer first. Sorry. Okay, so the work is titled Virus Resistant to Freezing-thawing Cycle. I hope you enjoy the talk. Let's start with a little bit of introduction. So the study of life in hard conditions is essential to astrobiology. Life on Earth can withstand challenging conditions. Extreme temperature, desiccation, acidic pH, or higher radiation levels are only examples. Hypothetical life outside Earth could develop in even more extreme environments. So the biology of these environments can help us to understand the condition under which life could emerge. One of the most extreme circumstances that exist in our planet is relevant differences between night and day, such as deserts, where cyclical episodes of freezing and thawing are common. Then the study of the possible adaptation is quite relevant. Next question would be why we use viruses in experimental evolutionary studies. So viruses are biological entities that are understood as chemical systems quite similar to the so-called primitive replicators that are capable of storing and transmitting information. They are composed of a genome enclosed in a protein capsid, and in the virion form can withstand more extreme conditions than most cellular life. A general property of viruses is their high error rate, which in the RNA virus reach values in the order of 10 to minus 6 of 10 to minus 4 errors per nucleotide copy. This high error rate, together with the usually large age of populations, lead to the generation of highly diverse populations that have a high potential for adaptation. For all these reasons, they are a usefulness tool to unveil the molecular mechanisms that allow adaptation and evolution of life, and we can study the evolution in real time. So in our lab, we use the Q beta virus as a model. Q beta, this is virus, is a bacteriophage that infects the bacterium Escherichia coli using the conjugative spirus as the receptor. It has a genome of single-strand positive RNA of 4,217 nucleotides. It has no envelope, and the genome encodes for 3 structural proteins and polymerase. These proteins are the A2 or maturaton you can find here in orange, the major capsid or protein in purple that do the capsid, and the A1 or minor capsid in pink that is produced for a ribosomal read-through of the genome. And you can observe this virus particle has between 6 and 7 copies of this protein, the A1. We choose this virus because it's genomes. For one side, it is a very small genome size, only 4 kilobases. And so any genotypic change can be seen very easily in the phenotype. And the genome is RNA, so it is more prone to mutate, more prone, it means that more prone to evolve and adapt into a new condition. So once we have the model, how do we do the evolution experiment? In this case, we begin with a wild-type population of the virus at a known concentration, and we put under the condition we want. In this case, the chosen condition is the freezing at minus 20 Celsius degrees, only the virus. And after 16, 18 hours, we thaw at 4 degrees in the fridge or in ice. We choose this temperature, minus 20 degrees, because the molecules of water form crystal and damage the material. Once we have the virus thaw, we allow them to reproduce, to replicate in the host in Escherichia coli at the standard condition. And after that, we try to know how was the damage. We try, like I say, no, but how was the damage? And later, from this, we took a part of them, of the population, and free and thaw again. This was on during 30 consecutive cycles, and we obtained 3 independently evolved lineage that we call it L1, L2, and L3. And we have to say that a control line was also established in which the ancestral virus was subjected to 30 transfers, but without the freezing and thawing cycles. So, firstly, to check whether or not this population had adapted to the new condition, we perform a freezing-thawing resistance test. In this case, we put a part of the population evolved, of the 30 transfers, L1, L2, and L3, and the cubed ancestor, and we free and thaw during 3 consecutive cycles without amplification in between. And as you can see here in the third, we found that the 3 populations have been adapted. L1 and L2 much better than L3, but L3 is still significant. After knowing the populations have been adapted to the new condition, the next step is wondering how. So, to identify the mutations responsible for the resistance, we use Sanger sequences to determine the consensus sequence of the 3 evolved lines. Don't worry if you are not familiar with this type of scheme. It is easy to follow. The first scheme here shows a representation of the genetic map of Q beta, and below there is an schematic representation of the mutations found in the genetic sequence of the 3 populations, L1, L2, and L3, and the control. The first notable observation is that of total of 11 mutations detected in the 3 lines, 8 are located in the structural proteins of the acid. From these, 6 are non-synonymous, 2 are synonyms. Non-synonymous mutation are mutations that change the protein sequence, and they have a much greater effect on an individual than a synonymous mutation. These mutations can be selected because they increase freezing thaw resistance, or because they increase the ability of the virus to replicate. The mutations, the 1312, 1930, and 2223, we know that for experience laboratory are frequent during the evolution of this value of Q beta. So maybe it's enhancing the replicate. And the remaining non-synonymous mutation of the acid proteins maybe is increasing the resistance to the freezing cycles. In conclusion, we can say that the viral population is giving stability to its shell to better cope this new situation. And as soon as we know the main mutations that are conferring this advantage, we want to know how these mutations behave particularly. So, for that, we do single mutants to study single mutations. Using the technique of site-direct mutagenesis, we make de novo viruses that contain only the desired mutation. We obtain the so-called single mutants. Firstly, we carried a freezing-thawing resistance test, but in this case with the mutant, the test is like the previous one. 3 consecutive cycles of freezing-thawing with amplification. And the mutant was this that I'm not going to say the numbers. You can see here in the graph the mutations 1494, 1665, and 2223 are the mutation, in this case, in the freezing-thawing cycles. In addition, we want to study if these mutations confer better virus replication, because, as I said before, are 2 selective pressures acting on the virus. For that, to test that question, we do a replication ability assay. And we saw, in this case, is the case of the mutation 1312, 201, and 2223. So we can say that there is a balance between mutations that are conferring better stability to the viral capsid to resist freezing and thawing, and mutation that, in another side, are improving viral production in the population. So finally, from this adapted population, we isolate biological clones that are individual viruses that came from the whole population, in this one, L1, L2, and L3. Biological clones correspond to viruses containing in isolated plate, which are generated from the replication of a single viral genome. And we do this because this population are very heterogeneous. This virus form the call it quasi-species. And in this way, we are analyzing how individual viruses behave in the same population. Like how I show you here is another freezing-thawing resistant test, but in this case, with the different isolated clones and the ancestor in black as a control. In the graph, the horizontal line represents the average of the 4 clones analyzed in each population. As you may see here, in the same population, there are viruses that have not adapted. It's the case of the clone 1 of the line 1 and the clone 4 in the line 2. However, as you may see, the majority of the clone in the population behave as the general population do. For this clone, we can provide an explanation because we sequence. I'm not going to show you the whole mutations that have these clones, but these atypical clones is because the one in red is because has the 201 mutation that we know is disadvantageous for the freezing-thawing cycle. And the other, the clone for the population 2, has not any mutations of the general. And that's it. To conclude, I want to summarize and read some clear messages because I know this is an evolution experiment, but a little bit of freezing-thawing cycles as a selective pressure that strongly reduce the infectivity of the virus. And after these cycles, viral populations resist this selective pressure better than the test. Important, most of the non-synonyms mutations are located in the coat in the A1 proteins. So we hypothesize now that the resistance is in the way to do more high stability of the viral acid. And finally, so there is a balance between mutations that are conferring better stability to the viral acid to resisting to resist the freezing and thawing cycles, and the mutations that are improving the viral production. Thank you so much. I would like to thank my team to support the Polish Astrobiology Society for giving me the opportunity to present our work. Thank you very much.